Hi everyone, it's the unqualified biologist here, and today we're going to talk about DNA replication. Now this series is going to be more focused towards O-level curriculum, but I believe that older students will find some of this content very useful as well. We're also going to talk about the Michelson-Stahl experiment, which was a landmark experiment done in 1958 by these two people called Michelson and Stahl. And what it did was basically revealed the mechanism for DNA replication. So what is DNA really made of? As you can see here, DNA is made of three key components. A nucleotide is the subunit, the basic building block of a molecule of DNA. And within this nucleotide, there are three main components. The first one is the nitrogenous base. That is going to be your ATCG. You also have the 5-carbon sugar. Now in this case, the 5-carbon sugar is deoxyribose, hence the name of DNA, deoxyribonucleic acid. And your third one is going to be this phosphate group. Over here, that's the one represented in green. And what's holding these things together? What holds each nucleotide together is the phosphodiester bond. This is a critical term you have to know, and what's holding each of these two strands of DNA together, each of these nitrogenous bases together, this is a hydrogen bond. Now this guy over here, this is Erwin Chargaff. What Erwin Chargaff did was basically he found out the ratio, these relationships that each of these nitrogenous bases have with each other. So here we can see I've drawn out the structure of DNA. And more about Chargaff's ratio. These are the four nitrogenous bases, and what he said was that A to T is in the ratio 1:1 and G to C is in the ratio 1:1. What this allowed him to conclude is that A is complementary to T, while G is complementary to C. And as we can see in the previous slide over here, A is bounded to T and G is bounded to C. Now in this experiment, he didn't have advanced technology, so the ratio looks something more like 0.99 to 1.01, which roughly translated to a 1:1 ratio when rounded up and rounded down respectively. But now modern genetic testing has shown that the ratio is actually exactly 1:1. Another thing you want to take note of is that there's another nitrogenous base called U. This is uracil. Now this one isn't found in DNA, it's actually only found in RNA or ribonucleic acid, which we will go more in detail when we talk about translation and transcription of DNA into proteins.